Good afternoon, everybody. I'd like to thank the organizers for inviting me. This is actually the title that was given to me, and in the spirit of the questions and answers, I think I'd like to pose the question to you, but maybe after you hear my talk. The topic I'd like to focus on are metastases, and the task that everybody, I think, would agree is very, very important, is to prevent, hold, retard, or cure metastases. And the question is, how do you do it? What do you focus on? Fogelstein and Kinsler, both being kind of gurus in cancer genetics, have written 8 years ago in Nature Medicine that although abnormalities of cancer genes are essential contributors to cancer, most abnormalities in these genes occur relatively early in the disease process, and none is a very strong word, and none are known to be associated with the metastatic stage. If not oncogenes and tumor suppressor genes, what? The answer was given 120 years ago by Stephen Paget, who wrote that when a plant goes to seed, its seeds are carried in all directions, but they can only live and grow if they fall on congenial soil. In today's nomenclature, the seed is the tumor, and the soil is the microenvironment. What's actually the microenvironment? Here you see a scheme of a solid tumor, any solid tumor, and you can see that in addition to cancer cells, there are quite many non-cancer cells in the microenvironment, some resident, some infiltrating. There are among the infiltrating cells, there are bone marrow-derived cells, macrophages, neutrophils. There are among the resident cells are endothelial cells, fibroblasts, and so on. Let me now go more or less in an ordered way in showing you what we mean when we talk about the microenvironment. As I told you, resident cells in the microenvironment, endothelial cells, some fibroblasts. If we are talking about infiltrating cells, we're talking about lymphocytes, macrophages, fibroblasts, and many others. And then there is many proteins in, for instance, in the extracellular matrix, such as collagen, fibronectin, and integrins. They're released molecules, released from both from the tumor cells as well as from the non-tumor cells in the microenvironment. Cytokines, chemokines, antibodies, proteases, angiogenic factors, other enzymes. Most solid tumors have or some regions in the solid tumors have a low oxygen pressure, hypoxia, and obviously if a patient is treated, drugs do find their way to the microenvironment. The point that should be remembered that these factors, all these factors, may interact with tumor cells and with non-tumor cells in the tumor microenvironment. The result of these interactions is a regulation, up or down regulation, or could be an up or down regulation of gene expression in these cells, and the outcome in many cases is a enhanced or tumor progression. The microenvironment is not only a thing to think and work about with respect to cancer research. Also other fields, for instance, botanists do work. Here you see the picture of 3 plants growing in 2 microenvironments, under or immersed in water and outside of the water. As you can see, the morphology of these 3 plants is completely different, whereas the genetic entity of these plants is obviously the same. Maybe gene expression is different. If we now go to the tumor level, here are these are cloned pre-malignant nevus cells grown in culture this experiment was done by a former graduate student of mine, grown on two different microenvironments, on plates coated with an extracellular a protein called laminin. The other cells were grown on fibronectin. The cells were stained for two different proteins, actin and focal adhesions. And as you can see, the organization of these two proteins is completely different depending on the microenvironment the cells grow on. And we did similar experiments also with in vivo experiments, and the metastatic phenotype of similar cells, identical cells, grown on two different microenvironments was completely different. These studies were obviously published. Now, this is probably the most important slide in my short talk. And this summarizes four main characteristics of tumor microenvironment interactions. We all have to remember that the tumor microenvironment is a very complex, a dynamic and ever-changing milieu. The tumor cells, as well as the non-tumor cells, change in the microenvironment as the tumor progresses. In other words, the tumor of today is most probably different from the tumor of 2 months ago, and different from the tumor later on. Another very important characteristic is that a certain microenvironment component could act completely different in an opposing way during the tumor progression. Probably the example for that is TGF-beta, when in breast cancer, for instance, in the early stages of the disease, it is an anti-tumor agent. In later stages, it promotes tumor progression. Then something very obvious, that the tumor microenvironment of the primary tumor and the tumor microenvironment of the metastatic sites are completely different. This is very obvious. I'm not going to elaborate. And then maybe very relevant to the talk, to this theme of my talk, is targeting the tumor microenvironment, that when after these two cells, one the tumor cell, the other one is a microenvironment cell, let's say an endothelial cells, do interact, they come in contact, both cells change their phenotype. And it is known, for instance, that the markers on the endothelial cells of the tumor are different from markers of endothelial cells in the same organ. This means, and this is actually the rationale, that if there are specific markers on endothelial cells in the tumor, then these biomarkers, these altered biomarkers, could be targeted. Very quickly, through various possibilities of targeting tumor microenvironment interactions. I'll give you 3 examples of drugs that their purpose is to inhibit pro-malignancy interactions. And I'm not talking about efficacy or not efficacy, just in principle, what could be targeted. Angiogenesis, as I'm sure all of you know. Another thing that could be targeted is growth factor signaling. And again, I'm not talking about efficacy. And then another thing that could be targeted is the interaction between inflammatory components and cancer cells. On the other hand of the picture, what also could be targeted are interactions that promote resistance or promote anti-malignancy interactions, for instance, immune responses. These are reviews that I wrote about the tumor microenvironment. I was just given 10 minutes, so possibly something has to be written about. There are many others. Finally, I'd like to invite all of you, especially those interested in cancer microenvironment, to participate in the 6th International Cancer Microenvironment Conference that is going to take place in Suzhou, China, November this year. The website is given below. I'd like just to tell you that the previous meetings were in Tiberias, Israel, not very far from here, the second one in Baden by Wien, then the third in Prague, Czech Republic, the 4th international meeting in Florence, Italy, the 5th in Versailles, France. The last one, we had about 700 people, both basic scientists as well as clinicians. So with that, I'd like to thank you very much for your attention. And again, thank you for the organizers for inviting me.